Okay, good morning, everyone. We're going to get started. Thank you all so much for joining us this morning. So the purpose of today is really to give you an introduction and a flavor to drug repurposing. So we've got two speakers with us. We've got Rick Thompson from Findacure and David Cavalla from Helix. And they're going to give you an introduction and then a more in-depth understanding to what drug repurposing is. So if you'd like to ask a question, then if you have a Google account, you can ask a question through the Q&A option to the side on your screen. Or for ease, you can just tweet at us at #drugrepo, you can see on the screen, and we'll collect all your questions, and at the end, once our two speakers have spoken, we'll have them start answering it. So without further ado, I'm going to hand over to our first speaker, Rick Thompson.
Okay, good morning everybody. We're really excited about today, and hopefully it's going to be a useful and informative session for everybody. I'm here really to give a general overview on drug repurposing, make sure everyone's on a similar page and got an understanding of the principle of repurposing and why it's advantageous and why we're excited about it. So as Flora said, my name is Rick and I am the Scientific Officer of Findacure. So Findacure is a rare disease charity. Now many people might say, you don't need to worry about rare diseases, they sound like they don't affect many people, but we'd say that couldn't be further from the truth. There are over 7,000 different rare diseases, and that affects roughly 350 million people worldwide. I think we'll all agree that's a sizable population. 400 of these different have licensed treatments. Sorry, apparently we're having some technical difficulties with the audio. Everything better now? Okay. Yes, so there are 7,000 rare diseases known in the world, and they affect roughly 350 million people worldwide, which is a very sizable population, something we really should be concerned about. Unfortunately, only around 400 of those have licensed treatments, which means there's a huge underserved market, if you like. Millions of patients out there without any treatment and with very little hope of receiving treatment because the rarity of their condition, little research goes into the disease. So Findacure's aim is really to try and address this problem. And our first major focus is on empowering patient groups, trying to help patient groups form, help to professionalize those groups, to give them the skills they need to interact with the key players in the rare disease world, those people who can help them ultimately deliver treatments to patients. We also work to campaign for a receptive research environment, and our main activities there tend to involve forging collaborations, making sure patient groups know one another and collaborate with each other, but also that they get opportunities to meet the pharmaceutical industry, biotechnology companies, academics and clinicians, all of whom can help them hopefully find new treatments for their disease. And that forms the third key aim for Findacure. We really are looking to facilitate the development of new treatments for rare diseases, and that's where I come in. You can see my delightful face here. As a scientific officer, I'm trying to set up a new drug repurposing program for the charity. So the key question then becomes, what is drug repurposing? At its most basic level, drug repurposing can be thought of like recycling. We're trying to reuse existing drugs and find new uses for them. Essentially, we're trying to make a more efficient use of the existing pharmaceutical catalogue that's available to us. Repurposing is essentially the act of taking a drug that's intended to treat one patient population and demonstrating its effectiveness or its efficacy in a completely different group of patients. And generally, we do that through means of a clinical trial. The reason repurposing has got such potential is because the pharmaceutical industry has been very successful. They regularly generate huge profits every year. We can see that over 30 or so new chemical entities, new drugs, are produced by the pharmaceutical industry annually. And if you look back through the FDA, that's the drug approval agency in America, we can see that since around the 1930s, they've approved over 1,400 drugs, which is a good wealth of material for us to work with. The pharmaceutical industry also invests very heavily in research and development, with some estimates saying as much as $70 billion is annually invested in drug research. And the spend on pharmaceutical products is huge annually. Over $100 billion was estimated to be spent on prescription drugs in 2015. So clearly they're doing a great job in producing drugs that are helping people and really creating a huge demand for their products. The pharmaceutical industry probably has some kind of problem, though. Essentially, it's the way in which they generate the profits from the drugs. It centres around patents. They need to have protection on the drugs they're producing. This gives them essentially a monopoly on producing new chemical entity. But that time is ticking on that patent. Okay, apparently we're garbled again. Can people hear me? Sound returning? Getting better? Okay. Apologies, guys. Yeah, so patents have a limited duration. And this is a positive thing in one sense because it drives pharmaceutical industry to innovate, to find new products. And this is essentially what delivers new treatments to patients. Unfortunately, it seems that the success of the R&D pathway is slowing down in the pharmaceutical industry. And furthermore, the cost of developing new drugs is increasing. So the graph on the left clearly shows a nice increasing trend in drug approvals since the 1930s till about the mid-1990s when it peaked, and we see quite a sharp decline after that, possibly with a recovery in the recent years. On the right, in the green line, we can see the cost of research spend for the production of drugs, and we see that's going up constantly from 96 pretty much to around the 2010, 2011 mark, where it seems to have stabilized. So reduced output but increasing spend is a problem for any industry. So we need to begin to look for new models to find new drugs. The de novo drug discovery is essentially the standard approach for the pharmaceutical industry, which is finding new chemical entities. And the approach they generally use these days is a mass screening approach, trying to look at a whole wealth of different chemicals and testing them en masse in a given disease model, a model system which replicates some of the biochemical properties of a disease. They pass thousands of different chemicals through this model and try and look for any beneficial therapeutic effects in the disease area, and hopefully come up with a series of candidate models which could act as a treatment. Mass screening was kind of hoped to accelerate drug discovery, and it's been used probably for the last few decades, but it just hasn't been the case. It really becomes why. We might be able to focus on the method. You could say that it's an inefficient approach to finding new drugs. Drug screening tends to be quite naive. We don't know much about the initial compounds. What was hoped essentially was that there'd be a huge number of hits. You'd find lots of potentially active compounds, but that seems not to have been the case. And this means the whole process is very costly and inefficient. Secondly, because of the naive screening, we don't know much about the safety or the effect of those drugs on patients or even on mammals. This means that when the drugs that are looking to be efficient in the chemical models are provided to a mammal model or to a human model, they often prove toxic. They might not be taken up successfully by the body, or they might fail to reach the target in the body. And if not toxic, they might still generate lots of very harmful or unwanted side effects, all of which can make them really ineffective as a therapeutic agent. Finally, the models themselves need to be good, and if they're not good, they might not accurately replicate the disease in humans. And this can mean that a promising effect in the lab, a promising result in an early screen, can have very little impact on the patient. And this type of thing can be very costly to the industry, as it means a large amount of time and money has gone into developing a drug. And when it finally reaches humans, we see a kind of effect that we weren't hoping for. Another issue could be levelled at the timing of the introduction of mass screening. Essentially, at the time when this approach was developing and the technology became available, most of the biggest, commonest diseases had off-patent drugs available. And this left two major options for the pharmaceutical industry. Firstly, they could try and find new drugs to treat those common conditions that were clearly superior to the drugs that were available. Now, this isn't necessarily very easy. Firstly, it can make it harder to pass your clinical trials because you don't need just to show a small improvement, you need to show a big improvement. Something that can convince people to try a new, more expensive drug. Another option might be to target drugs for less common illnesses. And this is a good thing in many ways, but the pharmaceutical industry probably have a lesser understanding of those illnesses, and there's certainly lesser research in those barriers, and it's also harder to find patients for those diseases. So without a more open looking, without a collaborative approach, that can often lead to poor trial design, and the trials themselves are less likely to provide the evidence that the industry requires to get their drug approved. So all this kind of highlights some of the issues with the traditional approach and might help to explain why we've seen a decline in pharmaceutical output. We think drug repurposing is another model which could actually help to fix this and is beginning to change this trajectory. There are two major pools of drugs we can look at for repurposing. And the first of those would be the pool of existing generic drugs that are available on the market. And these have got a wealth of data available about them. We call this specifically drug repurposing. Another pool you might look at would be the pool of shelved pharmaceutical compounds. So those drugs that the pharmaceutical industry have basically stored and shelved in their libraries. Things that have been trialled in humans and proved safe, but didn't prove efficacious or effective in their original targeted illnesses. And we call this type of project a drug repositioning project. You might say there's a third pool, which lies somewhere in the middle, which will be looking at those drugs that are currently available on the market but still under patent. All of these types have a huge advantage, and that is essentially you're working with what you know. And if you work with the drug you know, you're going to be having a project which is hopefully faster, development that's cheaper, and also it allows you to explore more, a different range of diseases, a wider range of diseases, because you know more about it in the first place. Essentially, we're removing de novo drug discovery, which is very expensive and requires a lot of chemical knowledge. We also know a huge amount about the safety profile and the side effects of the drugs, and that's particularly true if we're looking at generic drugs, which have a very long use in humans, possibly in lots of different doses and in lots of different diseases, which gives us a lot of knowledge about those side effects. This would hopefully mean that we have ultimately a reduced requirement for early stage clinical trials. Some of those phase 1 trials which investigate safety or pharmacodynamics might be less necessary or be less stringent, and you might be able to provide safety data based on existing evidence. What we also know is that there are known pathways of action with repurposed drug. We know something about its biochemistry. Because of this, we can hopefully have really good ideas about which different drugs can be used to be repurposed into a certain disease area. So this means rather than doing a mass screening approach, we can do a selective screening approach. We can screen likely candidate chemicals. Apparently it's garbling again. Has that returned to normal? Still not. Sorry. Oh. Has the sound returned to normal now? Still not there. Okay. Better? Okay. So, apologies. If we have a known pathway of action, this helps us target our repurposing projects, essentially. It means that we hopefully can find intelligent candidates for repurposing and, again, save on a huge amount of time and effort. Within the pharmaceutical industry, repurposing, targeting shelved assets is certainly on the rise, and this has been done often in a really collaborative way, which is great to see. Big pharmaceutical companies are opening up their shelved compound libraries and allowing academic institutions to look at them and suggest projects they might use for some of those drugs. If those are really meeting the needs of the pharma companies, they're often very supportive and enter into a collaborative project with those institutions, possibly providing funding and certainly providing research support. This means that those shelved compounds can go into early stage repositioning research, which might be generally academic-led, but with pharma input. And as things develop, we'd hope to move into a late stage development, ultimately going for approval. And all of these research stages are going to be collaborative, increasing pharma involvement as we move nearer to the point when a drug eventually will be produced and move onto the market. This works really well for pharma because the final drug has intellectual property. It's not been reached the market before. And that means it can be sold in a very traditional manner. With repurposing, things should be really positive because there's such a wealth of information on those generic drugs, which could make it very easy, or much easier to find candidate drugs for different diseases. However, there's certain problems in securing intellectual property on those drugs because they're already widely available and produced by a range of companies. This means that companies have to look into mode of use patents. So essentially saying they're allowed to use a given drug for a specific indication or specific illness. This should work quite well, but unfortunately they're quite hard to defend because it's possible that other generic drugs which are produced off-label for different indications might be prescribed by doctors into the new indication. And this is hard to detect and hard to prevent, so it can make it harder for big companies to return their investment. So there's somewhat less interest in repurposing and repositioning, although that again might not be always the case. Certainly because there are options for pharma. They can look at changing the drug in some way. So for example, they might try and increase the dose. They may also try and change the delivery method, so change it from a tablet to a nebulizer or inhaler, for example. Finally, they might change the formulation, so changing the chemicals that are in the tablet along with the active agent, which might help it to be delivered to a specific target within the body, for example. Now, all of these things can help the pharmaceutical company gain intellectual property, but they tend to require new safety data for use in humans because you have a new chemical that's been provided to the body. So in some ways, you're losing some of the advantages of repurposing here, but gaining the ability to protect the new drug. Repurposing without reformulation, this type of approach, can appeal to some pharma groups still. It definitely can appeal to academics and patient groups though, because we have a lower threshold for entry. Okay. So one really good example of that is nitisinone and the AKU Society. So the AKU Society is a patient group that's active in research, and they work on the rare disease alkaptonuria, which is also known as black bone disease. And this is caused by a lack of the enzyme that degrades homogentic acid in the body. So homogentic acid, or HDA, tends to build in the body, and this turns the bones black. But it also causes a form of early onset osteoarthritis, so it's really quite debilitating for the patients ultimately. There are lots of other symptoms and side effects as well. The AKU Society learnt about the drug nitisinone, which is a drug that's been previously approved for the treatment of tyrosinemia type 1, another very rare disease. Now, nitisinone also works on the pathway that produces HGA, affects the production of HGA. And what this means is if patients with alkaptonuria were prescribed nitisinone, it should prevent HGA build-up completely and therefore remove the symptoms of alkaptonuria. So based on this, the AKU Society have managed to raise funds, particularly using Horizon 2020 grants, which have allowed them to run their own clinical trials to test nitisinone for alkaptonuria. And the phase 2 trial is completed, and they're currently working on the phase 3 trial to try and prove the efficacy of nitisinone within the disease. At Findacure, we think this is a fantastic way of working, trying to have patient-led research, but it's a very hard thing to fund. So we're trying to develop a new funding mechanism called a social impact bond to help patient groups or academic groups in rare disease projects that are difficult to fund in generic drug repurposing. The bond's relatively simple. We are searching for private investment into the bond. With this money, Findacure hope to fund a number of clinical trials, phase 2 clinical trials in generic drug repurposing. What we're going to do is trying to target rare diseases that have a particularly high cost to treat or care for within the NHS, but without any real active treatment that can address the symptoms. So essentially we're managing symptoms in these diseases at a high cost. If we can show with our phase 2 trials that the drug is successful, it can be repurposed into that rare disease. We hope the NHS will then prescribe that drug off-label for the rare disease, as there's no other alternative. This will clearly save them money, as the drug will be generic, and also improve patient health. What we'd like to do is then access a proportion of those savings from the NHS to be paid back into the social impact bond. This is essentially a success payment, a payment for our successful trial. This will allow us, therefore, to return the initial investment, hopefully generate a return on the investment, and then finally, if we are successful enough, have money left over to invest in further clinical trials. With enough initial trial success, we could ultimately eliminate the investment side and have a self-sustaining system, essentially, which is allowing us to do a relatively constant level of generic drug repurposing in rare diseases. So this is the project I've been developing for the last few months, and the main thing I've been looking at recently is trying to identify repurposing projects. And there are 3 major strands for that which I can focus on. Something that's proved quite effective and is interesting for patient groups and academics alike is clinic-driven innovation, trying to talk to academics, researchers, patient groups, doctors who are treating the disease or working with the disease and get ideas from them based on their understanding or their experience of treating it, often sometimes with off-label drugs. And this can lead to interesting repurposing ideas. Secondly, we can explore data mining approaches. So essentially computer algorithms which can search the scientific literature for different ideas on the types of connections between diseases and biochemical pathways and illnesses. And many of these algorithms are very intelligent, and they can allow you to find ideas that aren't actually reported in any single paper. And this can give you quite new and innovative ideas for treatments. Thirdly, there are omic approaches, so approaches that look at the genomes or transcriptomes of patients and try to understand what the molecules are produced from the DNA and how we can correct the different signals from a given rare disease. And that's something that Helix are working on to a great level, and I'm sure David Cavallo will talk to you in much more detail about after this talk. So for now, I'll end my talk there. I thank you very much for listening. I'll hope that the sound will improve as we go on, and pass you on to David, who knows a huge amount about how we go about finding these projects. So thank you very much.
Thank you, Rick. Okay. Right, so I'm going to talk to you about Helix, which is a technology company with a social mission focused on drug repurposing for rare diseases. And we've trademarked the phrase rare purposing, which incorporates that motif of recycling, which Rick talked about. And we think that repurposing offers a variety of advantages for rare diseases, in particular being a method of R&D which is quicker, cheaper, and less risky for R&D. Actually, over the years, drug repurposing has been referred to in a variety of different formats. Some people talk about repositioning, some people talk about reprofiling. I tried to introduce the word or the phrase therapeutic switching. They all kind of mean the same thing in my experience, and the nuances between them have kind of been lost with multiple people having different views of it. Nevertheless, the reason why we're focused on rare diseases is because we think there is an absolutely huge unmet need. And Rick talked about the fact that there are over 7,000 rare diseases affecting a population of about the same size as the United States worldwide, and less than 5% of these conditions have an approved treatment. There are, fortunately, however, a number of new initiatives which have been set up by public groups like the NIH in America. They established a new division called NCATS, which stands for, I think, National Center for Advances in Translational Sciences. They have a very specific remit to focus on repurposing in rare diseases. eRare is an EU funding mechanism for rare disease repurposing. We believe at Helix very passionately in the alliances that we can form with charities, and we have an alliance with Cures Within Reach, which is an American organization which has set up a program called Cure Accelerator, and we also have a degree of collaboration with Findacure and are very interested in the work they're doing as well. So, in essence, drug repurposing is becoming increasingly popular in rare disease R&D, and this is just going to get more and more of a major strategic drive towards research and development in this area. So, you've heard about some of the statistics from Rick on the economics of pharmaceutical development. One of the key problems in the area is that not every drug that you start off investigating turns out to be an approved product from which you can sell and make money. This graphic here shows the number of drugs that enter into the preclinical phase of R&D is increasingly whittled down to from about over 20 at the preclinical stages down to 1 approved new molecular entity. And the success rate that you show here actually is an overestimate of the true situation, because prior to the preclinical stages you have a huge amount of research which often is undertaken in an academic environment, and it leads to the overall timescale for R&D being at least 10 and maybe 15 or sometimes even more years. And this timescale obviously bites into the patent exclusivity and makes significant commercial problems for pharmaceutical companies. The other problem is, in addition to the risk, in other words, the risk of failure, there is a very high cost of the studies themselves. The estimated cost of per new molecular entity which is introduced onto the market has been getting higher, and therefore the number of new medical entities, which is shown in this graph, per $1 billion spent on R&D, has been getting fewer. There does seem to be a little bit of a chink of light on the horizon. There is a suggestion that since about 2006-07, there has been an improvement in R&D productivity, but we're still at the improvement is a slight improvement, and we're still at a very high cost. So we've got to do something about this, really. And drug repurposing offers a way of improving the metrics of R&D efficiency. Effectively, what you do, very simply, is you combine old data on an existing drug with new biology, for instance, supporting the activity or potential activity in a new condition. And by combining those two things, you can create various shortcuts to the entry into clinical trials. So the discovery phase is reduced in time scale, and then the development can occur as normal. So this reduces the time, the risk, and the cost of traditional R&D. In terms of time, the discovery phase is reduced from between 4 to 9 years to between 1 and 3.5 years. The risk is also reduced. The probability of developmental success, going back to the slide I showed a couple of slides ago, is increased from about 10% to about 25%. Obviously, the safety risk is much reduced, but the efficacy risk, the risk of the product turning out not to be efficacious in the chosen new indication, is mostly maintained. But if you combine the time risk and the reduced time and the reduced risk associated with repurposing as a strategy, this leads through to massive reductions in the overall cost of a new product. We estimate that the traditional cost of a new molecular entity is about $1.8 billion. That can be reduced to about $0.3 billion using repurposing as an approach. There are patentable approaches based on method of use, but there's a particular advantage in rare diseases because there is additional exclusivity, which many of the regulatory agencies have offered to incentivize research into these conditions. So this slide here shows in a graphical way the alternative approaches to new products based on repurposing and based on the conventional discovery route. At the top, you can see that the number of you need to go through in the discovery part of the R&D process is fewer, and the time taken for that repurposing discovery to take place is significantly less than in the conventional NCE discovery approach. And this then leads through into an accelerated entry into clinical trials, but during the clinical trial process, the development process still takes a significant amount of time. So the take-home messages I want you to leave you with from this presentation are the following. Firstly, it's not an unusual thing for drugs to have secondary uses. In fact, I would say 90% of drugs have secondary uses, and that may be through a mechanism of action which is the same, or a mechanism of action which is different from the primary use. It's therefore a vastly underexploited area, and it can be very beneficial for rare diseases because sometimes the cost of R&D in a rare condition makes it impossible for a pharmaceutical company to make any money back except by finding a way of accelerating and improving the metrics for its R&D process. And repurposing obviously offers that. Thirdly, the dose, the route of administration, the duration of treatment, and the safety indeed could all be different. This is something which is, I think, underappreciated. And I'll refer to particularly the safety aspect later on. Repurposing is still risky for efficacy failure development. It's important, it's not essential, but it could be hugely advantageous to define a commercial impetus for the repurposing project. And as I've said before, orphan drug exclusivity is a major advantage for repurposing rare diseases. There are a variety of hypotheses which can arise from either computer predictions or from retrospective analyses of real-world experience of the administration of the drug in one disease in terms of its potential to affect a secondary disease. And I'll talk about that in a moment as well. The commercial impetus, you may say, well, why does a commercial impetus matter? Because if I know that a drug works in a secondary indication, why can't I just take it? Or why can't I get my doctor to prescribe it? Well, the reason why a commercial impetus is important is because if you can identify one, then the amount of data supporting the utility of the drug in a new condition is vastly increased. Clinical trials will be done, a massive information will be acquired, this can be inspected by regulatory authorities, and the safety and efficacy balance can be defined. Without it, generally speaking, the development will be very slow, and all the quality of data proving efficacy and the safety will be much poorer. And to emphasize that, I'd like to refer to a chemical known as acetyl salicylic acid, probably better known to all of you as aspirin, which was originally discovered and referred to by Herodotus 2,000 years ago as an extract of the willow bark. As an isolated pharmaceutical, it was first marketed by Bayer at the turn of the 19th and 20th centuries for pain, but then a further 70 years passed before the Nobel Prize-winning scientist Sir John Vane showed that it disrupted platelet aggregation and therefore for myocardial infarction and heart attacks, in other words, and stroke. It took a further 40 years before a widespread study was undertaken to show that aspirin not only did these things, but it also had a preventative effect on gastrointestinal and other cancers. In fact, actually, in something like pancreatic cancer, which is a very serious and problematic form of cancer, the chances of you developing pancreatic cancer, if you are already taking aspirin, are around about 25% of the chance of you developing it if you are not taking aspirin. However, despite all that evidence, there are no products containing aspirin to prevent the reoccurrence of cancer. It would be tremendously useful, in my view, if such a product was available and we had further information supporting its balance of safety versus efficacy. Bearing in mind also, of course, that aspirin does have side effects. It can cause gastrointestinal toxicity, and if we knew to a greater extent what that balance was, I think would be better off, particularly as aspirin can be used on a chronic basis and could be used between the first line treatment of cancer and the second line treatment. So in conclusion, new uses can take a long time to be identified, and then it takes a further substantial amount of time before they can be properly developed. There are, however, a number of successful examples of repurposing, and one which you may have come across and is a salient example is that of thalidomide, which was originally used for the prevention of morning sickness in pregnant women, and also used for prevention of insomnia. Many years after it was withdrawn for its well-known safety problems, it was reintroduced for the treatment of leprosy and secondarily myeloma. This is a fantastic example of clinical serendipity, which relied upon a leper or person with leprosy actually being given thalidomide by a doctor who thought it might assist in the patient actually being able to sleep, but then turned out almost miraculously to reduce the leprosy itself. Sildenafil is the generic name of the trademark drug Viagra, which was first approved for the treatment of erectile dysfunction. It's secondarily been introduced and marketed for the treatment of pulmonary hypertension, which is obviously a much more serious and lethal condition. So that's that. And then finasteride is a hormonal treatment for the reversal of prostate hypertrophy. It's also useful in the treatment of male pattern baldness. Obviously, both prostate hypertrophy and male pattern baldness are, if you like, examples of maleness, and finasteride does something to reduce the production of testosterone. Glycopyrronium is a very old drug for gastrointestinal ulcers. It's very recently been introduced as an inhaled treatment for chronic obstructive pulmonary disease. And there are many more examples which you can see at that URL given at the bottom of the slide. I mentioned earlier on about marketing exclusivity or exclusivity available through the regulatory process, which specifically applies for orphan diseases. And this was first introduced in the United States in 1983 and substantially increased the number of products which were approved by the FDA. So, for instance, over 300 products were approved since 1983, relative to fewer than 10 in the decade prior to the introduction of the Act. And because of the success of the Act, in terms of stimulating rare disease research and rare disease products, the main features of the Act were effectively replicated in most of the other sophisticated regulatory authorities around the world. There are slightly different hurdles and characteristics in terms of the prevalence of the disease, which is characterized as rare, and the different terms of market exclusivity, but the essential principle is the same, and that is that there are incentives for companies to enter into this kind of research in order that products are developed and that patients have options for their treatment. However, as I said, even with this exclusivity, rare diseases can often struggle to find a commercial impetus, and that's particularly where you have off-label prescriptions that can erode the returns to a company that has developed in this area. A few misconceptions. Just because you're recycling something doesn't mean to say that your products are going to be, if you like, rushed. There are a lot of groundbreaking drugs which have arisen from repurposing strategies. So there's something which actually arose in Cambridge, not far from where I'm speaking, which was originally called Campath. The generic name is alemtuzumab, and it was approved for chronic lymphocytic leukemia. A few years ago, some of the clinicians here started to be interested in the potential of this drug for multiple sclerosis, and it was ultimately approved a couple of years ago as a really effective treatment for multiple sclerosis. Pirfenidone is one of a bunch of 2 drugs which have recently been approved for idiopathic pulmonary fibrosis. It was originally a treatment for worms. Ketamine, you may have seen reports about this in the literature, is originally an anesthetic. It's a painkiller as well. To a certain extent, it's a drug of abuse, but it has absolutely remarkable effects in the treatment of severe depression, where patients who have been morbidly depressed for many years are almost instantaneously brought out of that condition by the administration. And I'll tell you in a moment about a drug that I've been involved with, which is espindolol, which is a variant of an antihypertensive, which has been repurposed for the treatment of cachexia. Another misconception is that large pharmaceutical companies are not interested. I mean, I would say they are perhaps less interested in this approach than conventional new molecular discovery, but they are involved in programs and projects around drug repurposing, and in particular, many of them have given access to some of their developmental compounds which have stalled in development, as Rick referred to earlier on, and made these compounds and the data from these compounds available for the academic sector to make proposals for them in terms of repurposing them and reinvigorating the development of them. And that's been supported in the UK by the Medical Research Council, and in America by the NIH in the form of NCATS, the National Center for Advancements in Translational Sciences. So I would say that it's overall repurposing is a major new strategy for drug rescue and pharmaceutical R&D, and almost month by month we see more and more reports of weird and wonderful things which have been discovered and offer hope to patients based on repurposed products. Helix effectively sits at the intersection between drug repurposing, patient charities, and the use of big data and omic analyses. So our approach, well, there are 3 approaches effectively. One is to take data on gene expression profiling, and we compare the expression profiles of drugs and the expression profiles of diseases. Secondly, we look at retrospective trial analysis to find non-predictive effects. And thirdly, I haven't got time to talk about this today, but a major new initiative which we're starting to publicize makes use of an algorithm to predict the right treatment for a right patient. The approach based on omic analysis effectively starts off with the gene expression profile of a disease and marries it to the gene expression profile of a drug. So what do I mean by that? Well, the RNA that you find in a diseased patient has normally certain increased genes and certain decreased genes which are associated with the disease. If we then compare that profile with the profile of the the genes which are expressed as a result of a set of drugs which are given to a cell, you can compare the ups and downs in one with the ups and downs in the other. The idea being that you reverse the disease-affected genes by administration of the drug. And then you can use algorithmic techniques to find the best drug out of a library of drugs to produce the effect you're desiring. And this algorithm was first published in 2007. It's been replicated and improved since then by a number of leading investigators worldwide, and indeed Helix has made its own contribution to this field. We are working using this approach with a number of rare disease charities. One is the NGLI1 Foundation. NGLI1 disorder is an extremely rare, ultra-rare condition which was only discovered a few years ago and has probably got less than 50 patients worldwide with that disease. The second one is a more common brain cancer which particularly affects children. Medulloblastoma is a cancer of the cerebellum and brainstem, and we are looking at identifying repurposing hypotheses for this condition as well, in conjunction with a charity called the APOD Foundation. In addition to the simple algorithmic approach which I've outlined earlier, we've refined these predictions using text mining, literature mining, and a database of real-world repurposing examples located at drugrepurposing.info, and we aim to produce accurate, protectable drug repurposing hypotheses, which are then validated in an experimental setting. We have a number of reports and examples of this validation, and if you want to find out more about those, please do get in touch. The literature analysis, the data mining analysis we employ is available on the internet, and you can use this for your own rare condition if you so wish at the URL which is given below, and it will provide you with literature examples and update you with contemporary literature reports as they evolve, so that you're kept up to date with what is happening in your chosen area. Now, I just want to talk a little bit about retrospective evidence. What do I mean by retrospective evidence? Well, in many cases, patients who are treated with one drug have that drug is found, by following the patient history, to have an effect on a condition which is not the intended target of the drug itself. And this is particularly the case in cancer, because of course in cancer any observed event where a patient gets cancer while being treated with a drug is always reported as a potential side effect. But if you look in a statistical sense at the incidence of cancer for patients treated with a number of chronically administered drugs, like say beta blockers or calcium channel blockers, HMG-CoA inhibitor refers to statins, and a whole series of other classes of drug. In these situations, the incidence of cancer which is observed for these patients is less than you would expect if they had been treated at all. And this suggests that these kinds of drugs are protective against the cancer occurring in the first place. So what I'm showing in this table are the cancer types on the left and the drug types on the left, and a variety of protective effects have been imputed for most of these drugs in most of these cancer types. But it's not just in cancer that you can look at this retrospective evidence. You could also see it in a variety of other conditions. And I'm going to give you a case history now about how this was used. This form of analysis was used to uncover a new treatment for cachexia. So what is cachexia? Well, it's It's the wasting disease which you get as in association with a variety of chronic illnesses, and in particular cancer itself. So on the right of this slide here, you can see a picture of Steve Jobs, who tragically died a few years ago of pancreatic cancer, and you can see how much weight he has lost in the later stages of his illness. It doesn't just come with cancer, though. Cachexia also comes with heart failure, a variety of chronic lung diseases, and a whole host of other serious conditions which are often end-of-life diseases. Cachexia itself is an independent risk factor for mortality. If you protect against cachexia, you can improve the outcome for patients with these various conditions. In cancer itself, about a third of patients, about a third of patient deaths are directly attributed to cachexia, and it's a condition which is complicated, and therefore, despite a lot of research into it, no widely approved agents are available. Therefore, represents an area of substantial unmet need. So, what was investigated was the effect of various beta blockers on the incidence of cachexia in heart failure. Now, the background to this is that beta blockers, you'll recognize them as antihypertensive agents predominantly, but they are also used and have been investigated for the treatment of heart failure. And heart failure is one of those conditions where cachexia occurs alongside the cardiovascular disease itself. And if you look at the occurrence of cachexia in heart failure, and you can see this in the yellow curve, where you have the number of patients with cachexia going up as the time period increases. Up on the bottom here, I've got 2 years of treatment. That risk of cachexia developing is reduced for patients who are treated with a drug called carvedilol. Now, carvedilol is a particular beta blocker which was ultimately approved for the treatment of heart failure. But you can see in this slide that it shows, in addition to benefiting heart failure, it also reduces the risk of cachexia. So what we wondered was whether all beta blockers would have the same effect. Now, people refer to beta blockers as a but actually the term beta blocker was coined before people really understood all the mechanisms by which they work. And beta receptors, which is the beta adrenergic receptor, was subtyped into beta 1, beta 2, beta 3, subsequent to the use of the word beta blocker in the first place. And in addition to that, other receptor activities have been uncovered for these drugs. So when we looked at a whole host of beta blockers, we weren't looking at a whole host of drugs which were the same. They were actually pharmacologically different. And of those about 40 beta blockers, only one of them was shown to have an effect on all of the aspects of cachexia in the hepatoma model, which is an animal model of cancer cachexia. So S-pindolol is actually a specific isomer of a known antihypertensive beta blocker called pindolol, and it produced better effects than the marketed product itself. And this is shown on this slide, where both against body mass and against overall survival, S-pindolol was highly effective and also more effective than an equivalent dose of pindolol. Upon that basis, a clinical trial was initiated where patients were given either placebo, a low dose of S-pindolol, or a high dose, and in particular looking at a primary endpoint of weight change over a 16-week period. And in that primary endpoint, the high-dose espindolol was extremely effective and extremely significantly different from placebo in terms of weight change. You can see these effects graphically in this slide, and over the 16-week period, the difference between the weight of patients treated with high-dose espindolol relative to patients who were treated with placebo is around about 4 kilograms. And this is a remarkable effect in a drug, in a condition which is otherwise extremely difficult to affect. In addition to weight itself, the body composition also changed, and there was an increase in the muscle mass, that's the lean mass, and there's also a small difference in the fat mass. Fat's quite useful in cachexia, actually, because it acts as a kind of a reserve to cope with situations of stress in the chronic condition. And finally, in terms of functional data, hand grip strength is one of those measures of the ability of the patient to improve in a functional sense. And again, high-dose espindolol produced a highly significant improvement in the strength of the patient's ability to grasp an object. Quite remarkably, also, there was no effect on blood pressure. This is in a way quite surprising, given the fact that the products are marketed as, but they're not particularly good antihypertensives. So in conclusion, I'd say that drug repurposing offers a radical way, a really radical way of reducing cost, risk, and time in pharmaceutical R&D. And for you as an audience interested in rare diseases, this, in my view, is offers substantial hope for improved therapies for your conditions. One needn't think that the characteristics of the original product are always carried over into the secondary indication or the secondary use. If you think back to thalidomide, as I said, it's remarkable that thalidomide was introduced for a secondary indication, given how unsafe it was recognized to be in its primary indication. And the fact that it's considered acceptable for the treatment of for things like leprosy and myeloma, points out that what we're really looking for in assessing a product is the balance of risk and efficacy. If you have a very serious condition, people will accept the fact that the product has a certain undesirable risk profile, whereas they wouldn't do it if you're just trying to treat a fairly benign thing like insomnia. And that's exactly the situation that pertains in thalidomide's case. I'd like to see more ways of getting commercial incentives behind repositioning products, because that gets pharmaceutical companies involved, it gets adequate amounts of data being generated, and it gets regulatory standard packages being developed. And fourthly, espindolol is currently the most effective developmental drug known for the treatment of cancer cachexia, and it is indeed a drug repurposing example. So that's me.
Okay, so thank you very much to David and to Rick for their presentations. Apologies again for some of the sound issues throughout. It might be linked to some of the internet connections that we have for the speakers. But we now, as we've come to the end of the presentations, move on to some of the questions that have been asked throughout. If you haven't had a chance to ask a question, please do either tweet at us using the hashtag #drugrepo, or do ask in the comments section of the webinar. So to begin, I'm just going to throw a question out there to Rick from Polly Moyer, who has asked a question earlier of if a drug is repurposed for a rare condition using the social impact fund process, will it still have to go through the NICE committee process? So Rick, could you take that on, please?
Aha. Can you hear me now? Excellent. Yeah, hi. Thanks for the question, Polly. The answer is, as usual, maybe, I suppose. As we are currently planning things at Findacure, we are looking to go for... can't hear me. Oh. Can you hear me, Flora?
Yeah.
Okay. Okay. Yeah, sorry. As we're currently looking to do things at Findacure, we're aiming to go for an off-label prescription model, which is essentially plausible within the NHS in those rare diseases where there's currently no treatment. However, basically in that route, we wouldn't need to go through an approval process. However, that isn't to say there aren't huge advantages in doing that. So with the right project and the right condition, with the right data, it would be ideal, I suppose, to try and go for a market approval for the repurposed drug itself. In that case, what you would expect is that the approved drug would then be submitted to NICE, where it would be assessed, and we would go through a health technology assessment to try and see if the drug itself is cost effective. So there's a range of different issues there, and it really would come down to a project by project basis as to how we'd like to approach it, and that will depend how things develop with our NHS collaborations.
Great. So we've got another question from the audience, which is from, hold on, let me bring it up. We've got a question of how can academia access to industry-shelved compound libraries, and what type of information, e.g. chemical structures, preclinical data, clinical trial results, can we expect to find? Can I throw that out to David, please?
Can you hear me? Yeah. So the calls that there have been, these are Research Council funded calls. They've been collaborations between, for instance, AstraZeneca and the MRC in the UK, and there's a wider section of pharmaceutical companies that was involved in the NIH NCATS collaboration. The way it works is that Research Council money or NIH money has been put into a pot, and the drug companies put their compounds into a pot or into a library, and then there are calls made for academics to make proposals as to the potential secondary use of these various compounds, and then money which is deployed will be used to validate these hypotheses. The companies make available the compound, and they make available the data for investigating the compounds, so particularly things like toxicological data or chemical synthesis data or formulation data, that sort of thing. In other words, things which can derive for development and then can be repurposed for the secondary development. The competitive nature of the call means that the academics don't necessarily get the money that they might bid for. It's done on a peer review process, and only one proposal can get funded for any individual product and individual call. So, but that's the way you would do it. So there have been these things in the past, and there will be more in the future. I do not think that there is currently one open that you can apply for, but if you want to get in touch, we can maybe point you in the right direction as far as where to look and keep up to date with the call process.
Great. So the next question is, are there any examples of the success from computational drug repositioning into an approved drug?
There are a number of publications positing new uses, and those have been making their way through the discovery and developmental pipeline. To my knowledge, it's a fairly recent technology, and it hasn't resulted in anything being on the... Sorry, can you hear, Flora? Yeah. There are a number of people who've investigated it, and hypotheses are being carried through, but it hasn't yet come out the other end in terms of an approved product. So I think it's too early to say on that.
Okay, and then a question to Rick. Have you already set up a social impact bond for repurposing drugs in the UK? Would it be for any rare diseases, or what areas are you currently planning to cover?
Hi there, sorry, my computer's lost connection, so I'll speak through David. We are in the process of developing the social impact bond at the moment. So it's a model that's been used in the UK and in the US actually for a number of different processes, particularly things like reoffending rates in prisons, helping to improve access to schools and for young people, that type of thing. There's one we know of in the UK. There's one health project that we know about in the UK, which is set up in the Newcastle region, I believe, looking at care for the elderly, but no other social impact bond is active in the healthcare field. So at this stage, we're in a developmental project where we're trying to prove the concept can work in repurposing. So we're trying to develop a series of financial models which can demonstrate that we can find high costs to the NHS for existing rare diseases, and also that the repurposing research itself could save significant funds. With that proof of concept, we can then go on to actually begin to work out how the social impact bond itself would function, and then begin to start the research. So it's relatively innovative and early stage at this point, but we are really open to different ideas, and we'd love to hear about any projects people feel that might fit with the model itself, so we can begin to develop that in collaboration with the NHS.
Okay, and then we've got a question from Deborah Mann, saying, given the concerns that are in place with regards to mitochondrial liabilities as an outcome of oxidative stress shown in many medicines today, what precautions are in place to ensure that this form of safety concern is addressed in drug repurposing? So I believe that's a question just about safety.
Okay, so the short answer is that repurposing is primarily a strategy for identifying new forms of efficacy of a drug rather than better safety. The drug is going to have a safety profile. The drug is going to have the safety profile which is not going to be changed by finding a secondary use for it. However, the balance between the sort of safety and utility of a drug can be influenced by the choice of indication, as I emphasized with the thalidomide example, where it was repurposed from a fairly benign situation for the treatment of insomnia into a much more serious situation where it was used for the treatment of leprosy and myeloma. And secondarily, I would say that by offering a more streamlined approach to new pharmaceutical R&D, you offer more treatment options to people, so that if you're faced with a situation where there is only one drug that affects that can potentially treat your particular situation, repurposing can offer a greater variety of products that might be effective, and some of those may have a better safety profile. But the drug itself, you're not going to be able to improve the safety profile by looking at an alternative condition. You can just change the balance between safety and utility.
Okay, great. So I believe those are all the questions we've received. If there are no other questions to be asked, then I would very much like to thank our two speakers for presenting this morning and now afternoon. And thank you very much for all of you who have joined in. I hope you found this useful. My apologies again for some of the sound and technical difficulties we've had. But I do hope that you managed to catch most of it. And we will be sharing the slides and afterwards, so if there's anything you missed, if you joined late, you'll be able to watch it again. I've just seen one final question that slipped through, so if the speakers are still okay to answer it, we've just received the question: will Findacure act as a market authorization applicant and take care of the pharmacovigilance? Could you quickly just respond to that question?
Yeah, sure. That's very much kind of to be decided. I mean, what we're aiming to do is build collaborations around each of these projects. So I would guess that in any project where we're looking to go in to a marketing authorization approval group, we'll be discussing that in detail with the clinical team, with the patient group team, and we'll come together as a group and decide on a project-by-project basis who's going to take various responsibilities. It's definitely something that we'd be willing to look into and help and provide support on if we didn't lead on. So it would come down to our collaborators' positions and our own positions for a given project as to how we respond to that most effectively.
Okay, and with that final question, I'm going to bring a close to the webinar. Thank you again so much for joining. Thank you to our speakers, and we hope to see you at a future event or webinar again. Thank you very much. Bye-bye.